Hello everyone, good morning all of you. This is Dr. Sudarshan Sambhajiraw Pedge, Assistant Professor Department of Zoology, Kailaswa Rasika Mahavidyalaya Devani, your zoology companion, and welcome to my lecture. So today we will learning about metabolism of carbohydrate. So let's get started today's lecture. First of all, what is meant by the metabolism? Question may arise. What is the metabolism? In simple terms, metabolism refers to all enzymatic reactions occurring in the cells. Okay, what is meant by the metabolism? Metabolism refers to the all enzymatic reaction occurring in the cells, that is called the particular metabolism. Okay, what type of enzymatic reaction is occurring in the cell? It includes all the changes taking place from the event of entry of substance, means mouth, right? Event of entry of substance into the organism to the discharge of the final end products in the outside through the outside environment. Okay, that is the total process is called as metabolism. Okay, metabolism is the process it obtains energy which is utilized for the growth, maintenance of life, and for the performance of biological functions. Okay, it is a useful process, and it will be the growth is depends on the metabolism as well as the maintenance of life. Okay, that is the one particular important, very important process of any living organism. Okay, so that is the one particular introductory idea about the or definition of the metabolism. Next is the one particular two types we have seen in the metabolism. That is the first is the intermediary metabolism. Generally, intermediary metabolism is the chemical reaction within the cells and tissue. Okay, as this chemical reaction proceeds in a stepwise manner through many intermediates, and the term intermediary metabolism is used, and the intermediates are called the metabolites. Okay, and that metabolites, it will be the helpful for the uh, building up of the body as well as the breaking down and functioning. Okay, this is one particular intermediary metabolism. Okay, so another second is the energy metabolism. Okay, in that process the overall energy production in an organism, it is simply the energy component of intermediary metabolism. Okay, wherein the chemical energy is derived from the degradative reactions is conserved to be utilized for synthetic process, and hence is also known as the bioenergetics. Okay, it is the one particular process, and that process it will be the helpful for the degradative reaction. Okay, it utilized the one particular synthetic process, and that synthetic process is also known as bioenergetics. Okay, this is the one particular energy metabolism of the types of metabolism. The second types, that is the energy metabolism. Okay, another next is the carbohydrate metabolism. Okay, now we will start our topic of syllabus. That is the carbohydrate metabolism. What is meant by the carbohydrate metabolism? What is carbohydrates? We know very well in the last lecture. Okay, carbohydrates, that is the mono, poly as well as disaccharides. Okay, so first of all, carbohydrates form the major bulk of human diet and also one of the cheap source of energy. Okay, and they are mainly in the form of polysaccharides as well as disaccharides. So we have studied in the polysaccharide. We have taken starch example, right? As well as in the disaccharide, sucrose, lactose, and maltose we also studied in the last lecture. Okay, so this is the one particular human diet. Okay, it's a very important human diet of carbohydrates. Okay, and that polysaccharides and disaccharides which hydrolyze into the monosaccharides such as glucose, fructose, and galactose by the enzymes of the digestive tract. Okay, the monosaccharides are absorbed by the intestinal villi and are transported into liver through the portal vein. Okay, so after the conversion of polysaccharides and disaccharides, monosaccharides it will be formed, and that monosaccharides it will be the absorbed by the intestinal villi and transported to the liver through the portal vein. Okay, this is the one particular carbohydrate metabolism. Okay, so now today we will start our carbohydrate metabolism in the first process, that is the glycolysis. In the glycolysis or EMP pathway, glycolysis is a process that results in the conversion of a molecule of glucose to two molecules of pyruvic acid. Okay, that is the one particular conversion of glucose into the two molecule of pyruvic acid. Okay, one particular, that is the process is called glycolysis or EMP pathway. Okay, and the term glycolysis is derived from the two Greek word. Okay, glycos means sugar, and lysis means dissolution, means the to break down. Okay, and this term glycolysis means the splitting of sugar. Okay, that's why we call it as that is the glycolysis. It means the glucose is one molecule of glucose is converted into the two molecules of pyruvic acid. Okay, that total process is called glycolysis. Okay, and it is also named as the embedded Markov of Parnas pathway after the investigation who made major the contribution to its discoveries in the 1930s. Okay, so this is the one particular total process is called the EMP pathway as well as glycolysis. That is, it means the glycolysis is the splitting of sugar. Okay, and in the glycolysis, glucose molecule, one glucose molecule is converted, or the conversion of two molecules of pyruvic acid. Okay, that is the one particular total process is called as the glycolysis. In the glycolysis, there are 10 major steps we have seen in that glycolysis process. Okay, first is the phosphorylation, second is the isomerization, third is the phosphorylation, and fourth one is the cleavage of fructose 1,6 diphosphate. Again, the second type isomerization is there. Okay, sixth is the oxidation and phosphorylation, seventh is the substrate level phosphorylation, eighth is the isomerization, ninth is the dehydration, and last 10th is the substrate level phosphorylation. Okay, that is the one particular 10 process we have seen in the glycolysis. Okay, after the ninth process, one particular two molecules of pyruvic acid it will be the form. Okay, so this is called the glycolysis, one particular pathway. Okay, so we see one by one step, okay, thoroughly. First of all, we have seen here in the picture of a particular cycle, and that cycle is the glycolysis cycle or EMP pathway. You have seen here glucose is converted into the pyruvic acid on various types of 10 types of steps is there. There is the one for hexokinase as well as the so many enzymes it will be included, or it will be useful for the process of glycolysis. Okay, so we will see thoroughly. First is the phosphorylation process. In the phosphorylation process, glucose is converted to glucose 6-phosphate, okay, with the help of hexokinase enzyme. Okay, and this is an irreversible reaction. Okay, which requires ATP and magnesium ions. Okay, that is called the total process is called the phosphorylation process. Okay, that phosphorylation process glucose is converted into the glucose 6 phosphate. Okay, another next is the isomerization. In the isomerization process, glucose 6 phosphate is converted to its isomeric form fructose 6 phosphate in the presence of the enzyme phosphoglucose isomerase. Okay, particular phosphoglucose isomerase, it will be the converted into the glucose 6 phosphate into the fructose 6 phosphate. Okay, that is the one particular. Second process is the isomerization. Okay, another next is the again second phosphorylation. That is the second phosphorylation reaction follows the isomerization. Okay, fructose 6 phosphate is the phosphorylated by ATP to fructose 1,6 diphosphate, and this reaction is catalyzed by the enzyme phosphofructokinase. Okay, you have seen here phosphofructokinase. It will be the used for the, or it will be converted into the fructose 6 phosphate into the fructose 1,6 diphosphate. Okay, and in that one ATP it will be the utilized. Okay, the first three steps of glycolysis form of first stage first where two molecules of ATP are hydrolyzed to provide necessary energy. Okay, so another next is the cleavage of fructose 1,6 diphosphate. Okay, in that steps you have seen here, the reaction involves the splitting of fructose, right? Splitting of fructose 1,6 diphosphate molecule between carbon atoms third and fourth by the enzyme of aldolase into two molecules of triose phosphate. Okay, namely glyceraldehyde 3 phosphate and dihydroxy acetone phosphate. Okay, we have seen in the last, so we have seen in the last um, figure. In that figure you have seen here is the dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. Okay, so this is the one particular cleavage of fructose. Okay, 1,6 diphosphate. Okay, with the help of aldolase enzyme. Okay, another next is the isomerization process. Fifth steps is the isomerization. Again, second isomerization we have seen here. There is the glyceraldehyde 3 phosphate is on the direct pathway of glycolysis. Okay, but dihydroxyacetone phosphate is not. Okay, in the last slide we have seen here two molecules, it will be the form, that is the dihydroxyacetone phosphate and glyceraldehyde 3 phosphates. Okay, but glyceraldehydes, it will be the direct pathway of glycolysis, but in the dihydroxyacetone phosphate is not. Okay, so it means the dihydroxyacetone phosphate, okay, dihydroxyacetone phosphate can be readily converted to into the glyceraldehyde 3 phosphate as both are isomers. Okay, isomerase. Okay, it means the glyceraldehyde 3 phosphate is an aldose and dihydroxyacetone phosphate is a ketose. Okay, this isomerization is catalyzed by the enzyme of triose phosphate isomerase. Okay, and this reaction is reversible, and thus the two molecules of glyceraldehyde 3 phosphates are formed from one molecule fructose, that is the 1,6 diphosphates. Okay, so this is the one particular process that will be the form the glyceraldehyde 3 phosphate. Okay, after that six steps is there. In the sixth step, oxidation and phosphorylation, it will be occur. Okay, glyceraldehyde 3 phosphates is oxidized and phosphorylated phosphorylations simultaneously under the influence of the enzyme of glyceraldehyde 3 phosphate dehydrogenase in the presence of NAD and inorganic phosphate to form a 1,3-diphosphoglyceric acid. The coenzyme NAD is in the oxidized form and has a net positive charge, okay, and hence is written as a NAD plus. Okay, here you have seen here the NAD plus. Okay, and this reaction is significant with the respect to energy transfer as the reoxidation of NADH to NAD, okay, by the electron transfer system uh, synthesis of one particular ATP molecules. Okay, so normally you have seen here that is the NADH through the electron transport chain. Okay, in the three molecules of ATP are formed in the three different sides of the electron transport chain. Okay, so this is the one particular oxidation and phosphorylation process. Okay, next seventh is the substrate level of phosphorylation. In that substrate level of phosphorylation, the high energy phosphate group in a 1,3-diphosphoglyceric acid or glycerate is transferred to ADP, resulting in the formation of 3-phosphoglyceric acid and ATP. Okay, so the enzyme that catalyzes the reaction of phosphoglycerate kinase in the presence of magnesium ion. Okay, the phosphorylation which occurs in this step, substrate level phosphorylation. Okay, in the next step, that is the eighth step, okay, isomerization process. In the 3-phosphoglyceric acid molecule undergoes the internal rearrangement to form 2-phosphoglyceric acid. Okay, and the phosphate group is transferred from the third carbon atom to the second carbon atom. Okay, this reaction is catalyzed by the enzyme of phosphoglyceromutase enzyme. Okay, and in the mutase is an enzyme that catalyzes the intramolecular shift. Okay, that is the one particular chemical group, it will be the shifted. Okay, that is the one. This is the one particular irreversible reaction. Okay, so another next step is the dehydration step. Okay, after the isomerization dehydration process, the 2-phosphoglycerate is dehydrated by the action of the enzyme enolase. Okay, with the help of enolase enzyme, the presence of magnesium and manganese to yield phosphophenol pyruvic acid, which has a high energy enolic phosphate group. Okay, phosphophenol pyruvic acid, it will be the form. Okay, after the dehydration or ninth step, last step is there, that is the substrate level for phosphorylation is the 10th step. In the final step, there is a transfer of phosphate from phosphophenol pyruvic acid to ADP. Okay, phosphophenol uh, pyruvic acid to ADP. Okay, thus a molecule of ATP is directly synthesized and pyruvic acid is also formed. Okay, this reaction is catalyzed by the enzyme of pyruvate kinase. You have seen here. Okay, that is the one particular of pyruvic acid, it will be the form. Okay, that total process is called the glycolysis. Okay, so today we will see here that is the one particular process it is called the glycolysis. First of all, we have seen in the introductory idea about chapter, that is the metabolism of carbohydrate. First of all, we have seen here that is the one particular definition of metabolism. After that, the type, that is the intermediary metabolism as well as energy metabolism. Okay, there are two types we have to see here. After that, the what is the carbohydrate metabolism and what would be the reaction, it will be the form here, or what will be the conversion of polysaccharide and disaccharide into the monosaccharides. Okay, after that we have seen today that is the one important process in the carbohydrate metabolism. That is the first pathway or first cycle, that is the glycolysis cycle. In the glycolysis cycle, we have seen here there is the 10 steps we have included. Sorry, we have studied here. That is the phosphorylation to the direct substrate level phosphorylation. That there are 1 to 10 steps we have seen today. So this is it for today. The remaining parts we will see later on. Thank you very much.